Hi, my name is Angelica Owen, and this is my individual project 1 on proteins. Protein structure and function. Proteins are the most versatile molecules in living systems and serve a crucial function in essentially all biological processes, with the ability to function as catalysts, transport and store other molecules such as oxygen, provide mechanical support and immune protection, and generate movement, transmit nerve impulses, and control growth and differentiation. Key properties of proteins. Proteins have several key properties which allow them to be so functional and versatile. Proteins are linear polymers built of monomer units called amino acids. The function of a protein is directly dependent on its three-dimensional structure. Proteins contain a wide range of functional groups such as alcohols, thiols, thioethers, carboxylic acid, carboxyamides, and a variety of basic groups. When combined in various sequences, this array of functional groups accounts for the broad spectrum of protein function. Proteins can interact with one another and with other biological macromolecules to form complex assemblies. The proteins with these assemblies can act synergistically to generate capabilities not afforded by the individual component proteins. Some proteins are quite rigid, whereas others display limited flexibility. Rigid units can function as structural elements in the cytoskeleton or in connected tissues. Limited flexibility may act as hinges, springs, and levers that are crucial to protein function. Synergistic, or various parts are working together to produce an enhanced result. Primary structure. Primary structure of a protein refers to the sequence of amino acids present in the polypeptide chain. Amino acids are covalently linked by peptide bonds. Each component amino acid in a polypeptide chain is called a residue or a moiety. By convention, the 1' structure of a protein starts from the amino terminal end and ends in the carboxyl 1 terminal C end. This is a diagram of a primary protein structure. Secondary structure. Secondary structure refers to local folded structures that form within a polypeptide due to the interactions between atoms of the backbone. The most common types of secondary structures are alpha helix and beta pleated sheets. Both structures are held in shape by hydrogen bonds, which form between the carbonyl O of one amino acid and the amino H of another. The backbone refers to the polypeptide chain apart from the R groups. And this is a picture of what a secondary protein structure looks like. Tertiary structure. The overall three-dimensional structure of a polypeptide. Primary, due to interactions between R groups of the amino acids that make up the protein. R group interactions. Hydrogen bonding, ionic bonding, dipole-dipole interactions, and London dispersion forces. Disulfide bonds are covalent linkages between the sulfur-containing side chains of the cysteines and are much stronger than the other types of bonds that contribute to the structure. Think of them as a molecular safety pin, keeping parts of the polypeptide chain firmly attached to one another. And again, here's a picture of a tertiary protein structure. Quaternary structure. Some proteins are made up of multiple polypeptide chains known as subunits. When these subunits come together, they give the protein its quaternary structure. The same types of interactions that contribute to tertiary structures, mostly weak interactions such as hydrogen bonding and London dispersion forces, also hold the subunits together to give them a quaternary structure. And here is a diagram of that. And this is a picture of all 4 structures. The first one is primary, secondary, tertiary, and quaternary. I did a slides on the protein insulin. Insulin is a small protein composed of 2 chains held together by disulfide bonds. Since these two chains are linked by disulfide linkage, insulin does not qualify as a protein with a quaternary structure. The amino acid sequence is highly conserved among vertebrates, and insulin from one mammal almost certainly is biologically active in another. Even today, many diabetic patients are treated with insulin extracted from a pig pancreas. Here's a picture of insulin. Biosynthesis of insulin. Insulin is synthesized only in beta cells in the pancreas in significant quantities. The insulin mRNA is translated as a single-chain precursor called preproinsulin. The removal of preproinsulin single peptide during insertion into the endoplasmic reticulum generates proinsulin. And here's a diagram of the biosynthesis of insulin, going from pre-proinsulin to proinsulin to insulin, with the removal of the C-peptide. Proinsulin consists of three domains: an amino terminal B chain, a carboxyl terminal A chain, and a connecting peptide in the middle known as the C-peptide. With the endoplasmic reticulum, proinsulin is exposed to several specific endopeptidases, which excise the C-peptide, generating the mature form of insulin. What does insulin do? Insulin allows the cells in the muscles, fat, and liver to absorb glucose that is in the blood. The glucose serves as energy to these cells, or it can be converted into fat when needed. Insulin also affects other metabolic processes, such as the breakdown of fat or protein. Problems with insulin. The most common problem associated with insulin is diabetes. Diabetes occurs when the body either does not secrete enough insulin or when the body no longer uses the insulin it secretes effectively. And here's a picture of a healthy pancreas producing insulin and a diabetic pancreas that cannot produce insulin. Type 1 diabetes occurs when the pancreas cannot produce insulin sufficiently to meet its own needs. It commonly occurs in children. An exact cause has not been found. Many consider it an autoimmune disease, and symptoms include tiredness, increased urination, thirst, and problems with vision. Type 2 diabetes, commonly associated with adults and lifestyle choices. People with type 2 diabetes will produce insulin, but often not enough to meet their body's needs. When the body does not produce or use it efficiently, blood sugar levels build in the body. The body also doesn't receive the energy it needs from glucose, which may cause fatigue. And here's a slide of all of my references.